Hello again. So we're going to continue on with our Chapter 6 information. So Chapter 6 really provides a pretty interesting overview of the different factors that affect the growth of microorganisms. And so now we're going to focus in on some environmental, or what are sometimes called physical factors that impact growth. Now, again, I think I mentioned in class when we started this unit, when you take anatomy and physiology, you learn about human metabolism. So all humans are alike in terms of their physical or environmental factors that are required for growth, and your chemical nutrients that are required for growth and survival. Microbes, though, on the other hand, microbes are diverse, very diverse. All right, so different microbes have different environmental factors that affect their growth. It depends on the environments that they're adapted to. The adaptations of microbes are very, very important. Different microbes are adapted to different environments. That means that their machinery that operates their metabolism is adapted to the conditions where they thrive. And as I have also mentioned in class, we're going to be taking advantage of this diversity of microbes as we go through your unknown bacterium identification project, because these differences are the basis for a lot of the traditional tests that are run and compiled to help you identify a microbe. You know, some organisms are going to be able to break down, this particular nutrient, and another group won't be able to. Group A has certain oxygen requirements that differ from Group B, etc. And so you start putting all those clues together, and that helps you identify organisms. Also, the human body, pretty large compared to microbes. We have different compartments in the body with different environmental conditions. And so you're going to have different microbes that either live there normally or can cause disease in different body locations depending upon the physical environments of your body. So what do we mean by these environmental factors? Microbes will differ in terms of their oxygen requirements, temperature requirements, pH requirements, osmotic pressure. So not all microbial cells are adapted to living in surroundings that have the same concentration of solutes. Some microbes, like if you're a microbe that lives in the Great Salt Lake, where you have a very high concentration of solutes in the lake water, the microbes that thrive in that lake have to be adapted to much, much, much saltier water than microbes that live in fresh water. Radiation tolerance. Microbes can differ. And radiation even includes visible light. So many microorganisms don't tolerate visible light well. And then other organisms, that gets more into the concept of biofilms that we've already talked a little bit about before. Many microbes change their behavior or even thrive better if they're part of a biofilm. And you've got to understand, if you want to culture microbes in the laboratory, then you've got to understand their physical requirements. You've got to try to reproduce the environment that they're used to living in if you're going to be able to successfully grow these in a laboratory on an agar culture medium or a broth culture medium that you use in the lab. That's very important, as you see here, for proper diagnosis. Diagnosis, again, that's figuring out what's causing a disease or a problem for a patient. Okay, so sometimes microbiology students are surprised to learn that microorganisms can differ in terms of their oxygen gas requirements. So not all microbes are like us. There are microbes that actually wind up being killed by oxygen gas, and you can read more about that in your textbook on this section. All right, so we require O2, that's oxygen gas. That's the form of oxygen that we use for our metabolism. And so do many microbes. Many of the microbes that we're studying, many of the microbes that live on or inside the human body or even cause disease, use oxygen gas as well. What do they use it for? They use it for a process called cellular respiration. That's not like breathing respiration. Cellular respiration refers to a complex process that cells use to break down nutrients like glucose and harness energy from those nutrients. And we're going to talk a little bit more about cellular respiration when we get into chapter 7. But what you see here on the slide, this is the overall chemical equation for cellular respiration. So you start here, C6H12O6, that's an input. These are our inputs. And over here, these are our outputs of cellular respiration. So C6H12O6, anybody know what that is? That's actually the chemical formula for glucose, a very important sugar nutrient that many living things love. And another input, for every glucose you're going to break down and harness energy from, you need 6 O2s, oxygen gas molecules. Your outputs over here, once you completely break down a glucose molecule, it's completely gone, you're going to end up with 6 carbon dioxide molecules. Now, if you notice over here, you started off with 6 carbons in that glucose. Output over here, 6 carbon dioxide molecules. So guess what? These 6 carbons that were in the glucose are now in CO2 molecules. You're actually going to make 6 water molecules for every glucose you break down as part of cellular respiration. Then as you break this apart, you break this bigger sugar molecule, glucose molecule, apart into these smaller pieces that you see over here. When you break bigger molecules into smaller ones, energy gets released. That's catabolic. So that energy gets captured to make ATP. And really a couple of inputs I should have over here. ADP, adenosine diphosphate, is an input. And then P with a circle around it generally refers to phosphate. So you're going to take an ADP and a phosphate and bond those together to make ATP. And ATP, again, that's the gasoline for the cells. That's where the energy is going to be stored so that you can quickly use that energy for things like active transport we were talking about on the last video lecture clip, or if a cell needs to rotate its flagellum so that it can move around. You need ATP to power those types of things. Heat over here is an output of cellular respiration as well, and that's because when you break down nutrients like glucose, energy gets released. Only some of it gets captured in the form of ATP. The rest of it gets lost in the form of heat. Same thing in our bodies. When you're exercising, you're using your muscles. Your muscle cells are doing lots and lots and lots of cellular respiration, and because of that, they're giving off lots and lots and lots and lots of heat. That's why you overheat when you exercise intensely. Okay, so interestingly enough, O2, wonderful. We got to have it to survive. So do many microbes. But oxygen gas has a dark, ugly side as well. It's very easily for O2 to get converted into other molecules that are chemically reactive and damaging. And some of these include, they have names you may have heard before, things like free radicals, singlet oxygen free radicals. You may have heard that. I've heard that on infomercials before. Superoxide ion, that's an O2 with a negative charge. Hydrogen peroxide, H2O2, the same stuff you got in little bottles that some people like to use as an antiseptic. And hydroxyl radicals, that's an oxygen and a hydrogen bonded together, and they have an extra electron, so they have a negative charge. So just as part of the normal metabolism inside cells, like they're showing you over here on this diagram, these types of things are byproducts of metabolism. Ultraviolet light rays from the sun hitting things like oxygen gas molecules will create some of these nasty chemically reactive oxygen-containing compounds. Cells will take these in from things like air pollution. We absorb these types of things. If you're a cigarette smoker, uh, cigarette smoking, you can absorb these things in from the cigarette smoke. So lots of things that we get exposed to, and just normal metabolic processes inside cells will generate these toxic oxygen-containing compounds. Okay, so guess what? These things are very destructive. So like they're showing you here on this diagram in the center, they will do things like damage your DNA. They'll chemically react with proteins. If your proteins get screwed up, they lose their three-dimensional structures. They're no longer going to work anymore, and a cell will eventually die. Incidentally, over here, this diagram that I've got over here on the right-hand side, where it says an antioxidant molecule is neutralizing a free radical. So free radical, that's some of these nasty oxygen-containing compounds are free radicals. Antioxidants are things that they're molecules that will react with these free radicals and kind of soak them up in a sense, or they neutralize them, they inactivate them so they can no longer damage your DNA and your proteins, or the DNA and the proteins of a microorganism. And so it helps prevent those types of nasty oxygen-containing compounds from being damaging to cells. So you hear about how you need more antioxidants in your diet, right? Many antioxidants are vitamins that you can obtain from fruits and vegetables. They tell you eat more fruits and vegetables so you can obtain more antioxidants in your diet. Well, this is a key reason why you need those types of things, because they help neutralize or inactivate the free radical type oxygen compounds that get generated in our own cells. And those types of things are true for microorganisms as well. All right, but on top of that, if you are any kind of organism, a microbe or a complex organism like us, if you're going to survive in the presence of O2, you have to have ways of destroying these evil oxygen-containing compounds. All right, if you don't, you're going to die pretty quickly. That's how rapidly these types of molecules are generated inside cells that get exposed to oxygen gas, and they can kill you pretty quickly. Okay, so what organisms that can survive in the presence of oxygen gas do? One reaction, so right here, you've got O2 with a negative charge. That's a superoxide free radical. There's an enzyme called superoxide dismutase. Enzymes catalyze chemical reactions, which means they speed them up. They're more likely to make them happen. You can also think of enzymes as just like the machines that carry out chemical reactions. What this enzyme does is it takes two of these superoxide free radical ions, combines them with two hydrogen ions that have a positive charge. Those are your inputs. The output of the reaction, hydrogen peroxide, H2O2, plus oxygen gas. Okay, so this enzyme makes this reaction happen. It forces these things to occur. All right, now once you've done that, H2O2, hydrogen peroxide, it's very reactive as well. So we're not finished with inactivating these things. These have to be destroyed as well. So there's another type of enzyme that many living things produce called catalase. And what catalase does is it takes H2O2 and converts it to water and oxygen gas. All right, so water is harmless. Regular O2 without a negative charge is not a harmful type of compound. So we have now successfully detoxified these oxygen-containing compounds. What you see over here, this is a test that you guys will actually run on your unknown bacterium at the end of our project. And you can take some bacteria and smear them onto a glass slide and drop some good old-fashioned hydrogen peroxide like you would buy at the drugstore on top of the bacteria. And if it bubbles, that means that your organism produces catalase enzymes. The catalase enzymes are doing this chemical reaction that you see right here. The catalase enzymes in those bacteria are converting the H2O2 to water and oxygen gas. So those bubbles you see right there, guess what those are? Those are oxygen gas bubbles that you see being released. Your cells do this too. That's why if your mama put hydrogen peroxide on a cut or a scrape when you were a kid and you saw all that bubbling going on, what you were actually seeing was your own cells, the catalase enzymes in your own cells, inactivating the hydrogen peroxide and converting it over to water and oxygen gas. So it probably had pretty limited impact on any microbes that might have been in your cut or your scrape. Okay, some organisms like us also have enzymes called peroxidase enzymes, and they do a different kind of chemical reaction. They take the hydrogen peroxide and just convert it over to water. But the take-home message from this is, if you are a living thing that lives in the presence of oxygen gas, you have to be able to detoxify these free radical compounds, these evil oxygen-containing compounds that are so destructive to things like DNA and proteins inside a cell. Otherwise, you're going to die. All right, so in terms of oxygen requirements, if you read through your textbook, different microbes can be classified in different ways according to their oxygen gas needs. So many microbes are in a group called obligate aerobes, and obligatory. Whenever you see aero in biology, that's usually referring to O2, oxygen gas. So if you're an obligate aerobe, you can only survive with oxygen gas. You're like a human. You've got to have the O2 to do your cellular respiration processes so that you can survive. And this does apply to many microbes, including some important pathogens. Pseudomonas aeruginosa is a bacterial organism that lives out in the environment, causes a lot of hospital-acquired infections or infections in patients with weak immunity. And good old Mycobacterium tuberculosis that causes TB, that's the bacterium that causes TB. It also is an obligate aerobe. It's got to have an oxygen supply. Incidentally, this nasty looking cut on the bottom of this person's foot, if you notice that kind of greenish color around the cut there, this is an infection with Pseudomonas aeruginosa. And some strains of that organism give off a green pigment. Remember, a pigment is a colored chemical compound. We saw those when you guys were looking at the organisms you grew on your Petri dishes after the swiping experiment. Some of those had colors to them, and that was because they produce these chemical compounds called pigments that have color. And Pseudomonas aeruginosa makes kind of a eerie looking green pigment. On the flip side, there are microbes that are called obligate anaerobes. So anaerobe here means without O2. It's obligatory that they live without oxygen gas. So these are actually organisms that die in the presence of oxygen gas. Why do they die? Because they can't inactivate your oxygen-free radicals. All right, those nasty oxygen-containing compounds, they can't inactivate those. So they actually get killed if they're in the presence of oxygen gas. All right, so believe it or not, there are quite a few obligate anaerobes that live in the human body. Most parts of the human body have an ample oxygen gas supply. It's delivered by the blood. That's the purpose, one of the main purposes of having blood and these blood vessels extending to all your tissues that you learn about when you take A&P. But there are anaerobic parts of the body, especially down in the intestinal tract, along the inner intestinal lining. You get down into the colon. That's an anaerobic environment. There's little, if any, oxygen gas down in that environment. And so 95 to 99% of the bacteria that live in your intestinal tract are actually anaerobes. There are some pathogens. Remember, we've talked quite a bit about the Clostridium species before the Clostridium genus, like C. diff, or Clostridium perfringens, which causes gangrene, Clostridium botulinum that causes botulism. Those are common environmental bacteria, but they are obligate anaerobes. So guess what? In the human body, they're not going to be able to survive unless they get into areas that lack an oxygen supply. So what you're seeing over here, this poor person has a gangrene infection, which is caused by Clostridium perfringens. If you had a severe injury, a burn, frostbite, the tissues die, which means they lose their oxygen gas supply because the blood vessels are no longer delivering oxygen gas to dead tissues. So if bacteria like Clostridium perfringens get into those dead tissues, they can actually grow in there. They get down inside those dead tissues and away from the oxygen gas in the air. They can actually grow in there and cause an infection even though they're anaerobic. And then Clostridium perfringens, unfortunately, produces a lot of nasty toxins, chemicals that are able to diffuse away from the area where they're actually growing and travel to other parts of the body. And these toxins can actually kill live, healthy, active tissues that are well away from where the bacteria are actually growing. That's why That's why gangrene is such a nasty problem, and sometimes they'll do an amputation to remove the infected tissues to help prevent that those gangrene toxins from spreading to other parts of the body and harming you. All right, so when you guys do your unknown bacterium project, one of the things you're going to test is the oxygen requirements of your organism. And some of you guys are going to have obligate aerobes. Many of you, though, are going to have bacteria that are in a category called facultative anaerobe. And it is facultative, it's not facilitative. So we were just talking about facilitated diffusion. This is a different word, it's facultative. And facultative comes up a few times in microbiology. It can be a little confusing at first when you're beginning to learn the terminology. Facultative anaerobes are organisms that prefer environments with oxygen gas. So they have all of the detoxifying enzymes that they need to get rid of those nasty oxygen-containing compounds that form when you live in the presence of O2. They prefer those environments, but they can survive without it. They have the machinery available to get by without oxygen gas. They can meet their metabolic needs without it. All right, a good example, good old E. coli. Many of the organisms that live down in your intestinal tract fall into this category as well. They love oxygen. They may grow better in the presence of it. Many of them do. But they can get by without it. So they're called a facultative anaerobe. Then there's another group, aerotolerant anaerobes. So they don't use oxygen gas. They don't need it. But if it's around, they can tolerate it. So they're not going to get killed by it, which means, again, they've got all these enzymes that will inactivate those nasty oxygen-containing compounds. Finally, there's a group called the microaerophiles, and based on the name, you can probably figure out what these organisms like. Micro means small. Aero, we said, is oxygen gas. A phile, like hydrophilic, water-loving. Phile means to love. So these are lovers of small amounts of oxygen gas. So in the atmosphere contains about 21% O2. These are organisms that require oxygen, but at lower levels, like 2 to 10% is ideal for them. So got to have it, but they can't tolerate as much as we generally have out in the open atmosphere, or even in most parts of the body. Most parts of your body will have higher oxygen gas levels than what these organisms like. A couple of examples, Heliobacter pylori, or H. pylori. You may have heard of this before. This is a type of bacteria that, believe it or not, can grow along the inside lining of the stomach, which, as we all know, has a low pH. That's an acidic acid environment. And these bacteria are able to shelter themselves from those low pH conditions. And so they're able to survive there. But also the oxygen levels are fairly low down along the stomach lining. They're down in that 2 to 10% range. And so because of that, H. pylori, that's a good environment for it to grow in. Borrelia burgdorferi, we've heard of this one before. This is the type of bacterium that causes Lyme disease. And it also prefers lower oxygen percentages. The Lyme disease bacteria like to get into places like your joints, into your joint fluids, where you have lower levels of oxygen gas. And again, these environmental preferences help dictate where these microbes grow inside the body. If you are a microaerophile like H. pylori, you're not going to be able to grow in parts of the body that have normal levels of oxygen gas. But you need some, so you're also not going to be able to grow someplace like the lining of the colon where there is little to no oxygen gas available. How do we characterize oxygen requirements in the laboratory? And you guys are going to run this test on your unknown organism if it ever quits snowing, and if we ever quit having additional holidays from school, we'll finally get to that project. But what you do, there's a special type of broth fluid that has certain chemical compounds in it that help maintain high oxygen levels at the top of the broth, low oxygen levels down deeper in the broth. So you set up a gradient of oxygen where higher levels up here, and it gets progressively lower as you go down toward the bottom of the tube. All right, so then you take an inoculating loop, you inoculate this broth with your bacteria, you incubate it for a few days, and you come back and you look at it. If your organism is growing only up here at the top where oxygen gas levels are high, then you have an obligate aerobe. It must have O2 to survive. If it's only growing down at the bottom where there's little to no oxygen gas, then you've got an obligate anaerobe. You must grow without oxygen gas. If you have a facultative anaerobe, remember we said that's an organism that prefers oxygen gas but can survive without it. Typically what you see is heavier growth up at the top and lighter growth down deeper in the broth where you have little to no oxygen gas. So most of these organisms they'll grow faster with oxygen, but they can still get by without it. So you won't see the growth won't be quite as heavy down deeper in the tube. Then if you had an aerotolerant anaerobe, so we said these are organisms that don't need or use oxygen gas, but they can tolerate it. They're able to detoxify the damaging oxygen-containing compounds. So those will generally grow pretty uniformly throughout a tube of this type of broth. All right, now when you actually run lab tests, they generally don't look like the textbook diagrams. This is more like what you would see in the lab. So if you look at tube number 1 here, notice you got a ring of heavy growth up at the top, so that's an obligate aerobe. Both of these in the middle are different kinds of facultative anaerobes. So if you notice like this one, growth is heavier up here at the top and lighter down here at the bottom. So that's like what we were talking about over on the diagram. This one is still a facultative anaerobe, but you can see it actually has heavier growth down toward the bottom. And again, that's more, relates to the diversity of microorganisms. Different facultative anaerobes are going to be better at growing without oxygen gas than others. This one over here is actually an obligate anaerobe. Notice how it's clear up at the top. So it can't grow where there's O2, but down here deeper where oxygen gas levels are little to none, it's able to thrive. So that would be an obligate anaerobe in that particular tube. All right, you probably will, working in a medical setting, have to see how anaerobes are cultured in a laboratory. So if you collect a sample from a patient and it's suspected that an obligate anaerobe might be causing the infection, you can't just stick that in the incubator like the incubators we have in the lab, because those incubators have lots and lots of O2, oxygen gas, in them. Anaerobes have to be cultured inside special containers like you see over here. Here's some Petri dishes that have been placed inside this container. And then you open up a gas pack over here that lets out CO2 and hydrogen gas. That's designed to replace the oxygen gas that would be located inside the chamber. And then over here, these palladium catalyst pellets absorb and react with the oxygen gas inside that chamber and deplete it. All right, so this is an oxygen-free chamber. So it's important to know from a clinical standpoint, if you're collecting a sample from a patient and it has to be cultured under anaerobic conditions, then you can't just stick those plates or those tubes or whatever they are into a typical incubator. You've got to use an anaerobic incubating chamber or a gas chamber like you see on that particular diagram. Okay, I'm going to halt this video clip so it doesn't get too super long, and then we're going to continue on with talking about some additional environmental factors that can impact the growth of microbes.